So Mendel was really one of the major contributions of Mendel was this idea of segregation and independent assortments. We're going to kind of go back to that and look at how that applies in terms of cell division and what's actually happening in the cell as predicted by those two laws. So in terms of segregation, this can be explained by the segregation of homologous chromosomes during meiosis. That means that we're basically, if you think about it in terms of the alleles, if you're thinking just in terms of alleles, we're talking about the ability of one allele to go into one gamete and a second allele off of a homologous chromosome to go into a second gamete. Now, Mendel's law of independent assortment also comes into play here, and that can be explained by the observation of the random alignment of bivalents during meiosis. And I would ask you to think about whether that's 1 or 2. To think about whether, or to determine whether it's 1 or 2, you have to remember when bivalents actually occur and know what bivalents are. Okay, so let's look at how segregation works. So here we have a cell that's going into meiosis 1. We've got our sister chromatids, each individual chromatid of a homologous chromosome. So this chromosome is homologous to the second chromosome. And we can say that this is a maternal origin and this is a paternal origin. And then we have our sister chromatids. Here during metaphase is when we get the formation of the bivalents. Because we're only dealing with one chromosome, we can't really see how the law of independent assortment is applied here, but we're just going to focus on segregation. Now, at this point, these are going to separate into these two new cells that are formed. The homologous chromosomes are segregating from each other or moving into to separate poles and then ending up in two different cells. Now, as we move down here into meiosis 2, the products of meiosis 2 are haploid cells. So now we've got the maternally inherited chromosome here and the paternally inherited chromosome here with its two sister chromatids, or its copy. Those are going to segregate again into two different cells. Now this ultimately ends up being our haploid gametes, where we can think back to our transmission genetics and our Punnett squares. These are what we would be putting along the outside of the Punnett square. So here and here and here. This representing the gametes here, and this potentially representing gametes here, if we were dealing with two separate individuals. But you can think of it that way. So in all of these cases, what we're having happen, or what's occurring, is actually segregation into separate cells. And by design, they're segregating so that the original, that these gametes end up with one copy of each of these homologous chromosomes that we started with in meiosis 1. Now let's talk about the law of independent assortment and identify where we can see independent assortment. Now this figure differs from the last in that here we're starting out with two sets of chromosomes. Here are in red again are our maternally inherited, and in blue are our paternally inherited chromosomes. This is in the cell of a new individual. Now in here there's two possible arrangements of these chromosomes during metaphase. And in terms of the arrangement, it's the side, if you will, that they're on. So if we look at this one, we've got these 4 chromosomes, 2 of which are homologous, and the other 2 are also homologous. As indicated by the Y gene, we'll call that the Y chromosome. No, let's not do that because that gets confusing. Let's call it the Y gene, and then down here we have the R gene on this set of homologous chromosomes. So these can line up where along this metaphase plate where all of our paternal chromosomes are on one side and then all of our maternal chromosomes are on the other side. Now again, we're going to go through and these will divide and produce these two cells at the end of meiosis 1. We are at metaphase again and we're lining up. Notice that we're still maintaining the same kind of orientation that we had up here at the top. So these alleles are then going to segregate into the haploid gametes at the end of meiosis 2. The same thing is happening here, where we've got a big Y allele on this chromosome ending up in this gamete, and a big Y over here ending up in this gamete. The little R alleles are going to segregate again, so that we get haploid cells at the end of meiosis 2. Now, again, since we have two sets of chromosomes, there's two possible arrangements of these chromosomes along that metaphase plate. We've seen one where the paternal and maternal are all aligned. That does not have to always happen that way. You could also get this scenario over here where we've got a mix. We've got the one paternal and one maternal, one paternal and one maternal on the opposite side of that metaphase plate. So now we're going to end up with a little bit of a different starting point here at the beginning of meiosis 2. So at the beginning of meiosis 2, we're going to see these lined up where we've got here's a paternal gamete, here's a maternal gamete, and here's a paternal gamete, and here's a maternal gamete. Notice too that means that we're going to get different arrangements of alleles in our gametes at the end here. So here we've got all the dominant alleles in the two gametes. We've got a little R and a big Y, a recessive and a dominant allele in the two gametes. At the end of this meiosis 2, after starting here, we're going to get two recessive alleles in two gametes and two dominant alleles in the second gamete. So now imagine that the more chromosomes that you have, the more possible arrangements you're going to get that are likely. So we can think of that as a math problem. We can raise 2 to the n, where n equals the number of chromosomes in a set. So that's basically the haploid complement. So we could get then for humans, where we have 2 to the 23rd, that should equal approximately 8 million different combinations of gametes that can end up, of alleles, and chromosomes that will end up eventually in these gametes. So this is a huge source. Independent assortment is a huge source of variation on its own. Now you can think of other sources of variation too, that being recombination, which even further complicates this process in terms of mixing or creating new arrangements of alleles on genes.